right, thank you all very much for attending the talk. My name is Sam Scarpino. I'm currently an assistant professor in the Mathematics and Statistics Department at the University of Vermont, although I'll have an update about that at the end. And I'm going to talk about how Coevolution facilitates influenza antiviral resistance evolution. If you're paying attention, you might notice that a couple of the words are missing. I'll explain why that is when we get to it. Very quickly, if you heard me talk last year, or if you're interested in gene co-expression networks, the paper describing our results is out in Proceedings B, and the code, etc., for the methods are all online. The bulk of the work I'm going to talk about was done by my master's student, Devengo Kale. He's moving to the University of Georgia to do his PhD. And then there was some earlier work that I did with Sean and Katya Cole at Duke. So there are a couple of interesting patterns in terms of the evolution of antiviral resistance in influenza that we want to try to explain both with some mathematical models and with some available data. And the first pattern is that we see rapid within-host evolution of neuraminidase inhibitor resistance. So I'll tell you what neuraminidase is in a second, but very quickly, it's one of the two key surface proteins for flu, and it's responsible for exit from the cell surface. So if neuraminidase is inhibited, then the virus is bound to the outside of the host cell and it will be degraded. So what we see, this is for H5N1. Each line here is a different patient. This is day since admission, and this is a measure of viral load here, so higher is a more severe infection. And these individuals in red here succumbed to the infection and died, and they were all infected at the end of time with oseltamivir resistance as neuraminidase inhibitor resistant strains, but these strains were all sensitive at the beginning when they were admitted to the hospital. We also see interesting patterns of community spread of antiviral resistance. These are data from Japan in 2013, and during the first outbreak of this strain of H1N1, there was no observed resistance to oseltamivir in the population, and then during the second wave here, a year later, the light blue here are resistant strains, the dark blue are sensitive strains, we see more resistance showing up in the population. And this tends to be the general phylogenetic pattern associated with the prevalence of antiviral or neuraminidase inhibitor resistance. So these are data from H1N1 pandemic influenza. This is a gene tree from the neuraminidase gene. And what we can see in 2009 is a single red branch. So there's only one lineage here that's resistant to oseltamivir. And then in 2010, we have more branches that are resistant to oseltamivir, more in 2011, more in 2012. So there's sort of steady creep in the prevalence of oseltamivir resistant influenza. Now, putting sort of this result together with this phylogenetic result, it sort of raises an interesting question, which is we see very rapid within-host evolution of antiviral resistance, and for the most part, it's due to a single amino acid change. And we see sort of a steady increase in the prevalence of resistance, which would maybe suggest that there's some selection pressure that's moving this resistance allele towards fixation, but for the most part, we don't really use antivirals. And the only people that typically get antivirals are sequestered in hospitals and not transmitting. So there's a sort of open question, which is, why are we seeing this increase in antiviral resistance, given that we're not really using antivirals? And as I'll show you in a second, there's a pretty extreme fitness cost to the virus for harboring these mutations. A study by Chow et al. Demonstrated this a little bit more convincingly. So this is the cost of resistance, and it's a little bit confusing because it's 1 minus the cost, so negative numbers mean there's actually a benefit to the resistance. And the proportion of cases that are treated, so this would be like 4% of cases treated, and then what these bands are the number of years for a resistant strain to rise from 1% to 50% prevalence. And this is where they think the empirical data are sitting. So for what we're seeing is, in fact, that there has to be some selective benefit to this resistance mutation, even in the face of very, very low fractions of antivirals being delivered to individuals. So very quickly to explain the life cycle of influenza as it's going to relate to our model is, so this is an influenza virus. It has hemagglutinin surface proteins in green and neuraminidase surface proteins in blue. And the hemagglutinin proteins are what's responsible for binding to the sialic acid receptor on the cell surface and triggering uptake so that the virus can replicate. And then you have budding off of the cell surface. And as these viruses bud off the cell surface, the hemagglutinin sticks back on again. It's the neuraminidase that cleaves the virus free so it can go on and replicate. One of the things that happens is that the host immune system will bind antibodies mostly to hemagglutinin and prevent binding and uptake. And what we tend to see experimentally and in data is that there is an interaction between the host immune system, which tends to favor higher binding avidities in hemagglutinin, that have to be compensated for by more active neuraminidase. So the host immune system selects for stronger hemagglutinin binding, and in order for the cell to exit, you have to have more effective neuraminidase to cleave that free. A paper by Handel et al. Developed this in terms of a mathematical model, which basically asked the question, how sticky should a virus be? Which is, if you have entry and exit, and the strength of binding that triggers entry has to be matched by the activity of the cutting molecule in order to trigger exit, that there's going to be coevolution between these two surface proteins. And again, if you're interacting with the host immune system, all three of those things are going to be sort of driven to some optimal, where the virus can maximally evade the immune system by strongly binding to the host cell, but then can still be cleaved free from the cell surface. The way neuraminidase works, again, is that you have viruses that are leaving, and then the neuraminidase inhibitor comes in and sits down on top of neuraminidase and prevents the viral release. So what we did is we took this model of within-host evolution of hemagglutinin and neuraminidase with the host immune system, and we embedded it in a population-level model where the influenza virus is moving through populations of humans over the course of multiple seasons, such that we're getting this slow buildup of antibody prevalence within the population. So we're going to have multiple waves of influenza moving through populations, and as flu moves through the populations, we'll get an increase in the relative number of individuals that have been previously exposed. And we'll ask what happens to these surface proteins. So again, we'll have maybe populations with higher average immunity that viruses are interacting with, and lower average immunity that viruses are interacting with. And what we see in terms of this resulting model, in terms of the fitness landscape that comes out, is this trade-off between neuraminidase activity and hemagglutinin binding that's modulated by the strength of the host immune system. So what we're looking at here is neuraminidase activity on the x-axis. So this is a more active neuraminidase that can cleave more strongly binding or higher binding avidity hemagglutinin. Here is the hemagglutinin binding strength. So again, these are going to bind more tightly to the cells, and in experimental studies they've shown that this is a mechanism for evading the host immune system. The darker the color, this is going to be the higher growth rate in the population as it's moving through this set of hosts. So darker blue means more fit in terms of the virus at the population level, in terms of the number of viruses that are spreading. And what you can see in the low immune host is one that on average the viruses are all doing much better, which is what you might expect, but the sort of optimal combination of neuraminidase activity and hemagglutinin is relatively weak binding and relatively weak activity. Whereas in the high immune host, we see this shifted substantially up and to the right, so that you have much stronger binding avidity being favored and much more active neuraminidase activity. And there's some very nice experimental evidence for this that was published in Science by Hensley et al. So what they do is they passage the virus either through vaccinated mice or naive mice, and they look for changes in hemagglutinin and neuraminidase, and they see 3 different changes in hemagglutinin that show up. And then they measure the binding avidity of these 3 different changes, and they show that these 3 different changes are associated with significant binding avidity. I'm not showing you the results here, but they also demonstrate that this correlates with immune evasion capacity of these different viruses. In our model, the effective antivirals is sort of what you would expect. So this is the fraction of high immune hosts in the population. So this would be all hosts previously exposed to the virus. This would be no hosts previously exposed to the virus. This is the population level effective growth rate. So this is the number of secondary infections from a single infected individual. For no antivirals, weak antivirals, moderate antivirals, and strong antivirals. And so what you can see is that as the antiviral strength goes up, the growth rate of the virus goes down. Papers by Bloom et al. And Butler et al. Were actually able to quantify what the cost was in terms of the most common resistance mutation. And what the most common resistance mutation does is it affects the ability of the neuraminidase glycoprotein to be presented on the viral surface. So these viruses end up with about 50% fewer functional neuraminidase on the viral surface. And so we're going to include this cost directly into the model, such that resistant viruses now carry 50% less efficacious neuraminidase. And what we see when we do this is the proportion of high immune hosts, again from none to 100%, and you can't see this here, but this is the population level growth rate. For weak antivirals, the sensitive strains are always outperforming the resistant strains. For strong antivirals, maybe not surprisingly, the resistant strains are always outperforming the sensitive strains. However, for moderate antivirals, what we see is that when you have low proportion high immune hosts, the sensitive strains are winning, and when we get to higher proportion high immune hosts, then the resistant strains are winning. And what happens in the model is that the hemagglutinin is evolving with the host immune system, higher binding avidity. That gets compensated for by more active neuraminidase. And those more active neuraminidase compensate for the cost of the mutation, which is fewer neuraminidase on the cell surface. So you end up with 50% less efficacious neuraminidase, but twice as functional for the ones that are actually on the surface. And it turns out that this has been observed empirically, so I won't read the whole title to you, but a very nice paper by Butler et al., where they were looking at these mutations showing up in influenza, found that in fact it wasn't that the neuraminidase resistance was being compensated for, so you didn't get neuraminidase resistance mutations showing up and then compensation, that in fact it was permissive mutations, that the mutations that were needed to compensate for the cost had already fixed in the population, and then you get this steady rise of neuraminidase inhibitor resistance. So what Devin wanted to do was to try to test this computationally. So we downloaded 1,500 influenza viruses that have been tested for neuraminidase inhibitor resistance by the CDC. We then used a tool to computationally predict the binding avidity from the side chains on those neuraminidase, and then we're going to include that in a model of evolution and ask whether we actually get a switch in binding avidity that's associated with the switch in resistance. And so the paper that we used for the estimation was published a few years ago. So here's what we find. These are our sensitive viruses, these are our resistant viruses, and this is the binding avidity of the hemagglutinin. And we see significantly higher binding avidity associated with resistant viruses in our sample. Now again, we computationally estimated what the binding avidity was, and so the only sort of experimental validation of this comes from that paper. We can put this onto a phylogeny, so here's the phylogeny of the hemagglutinin protein for viruses between 2005 and 2008. The colors here are either resistant or sensitive, and then we'll fit different evolutionary models to this. So this could be the evolution of the binding avidity of hemagglutinin, either Brownian motion or an Ornstein-Uhlenbeck process where there's some restoring force to an optimum. And then we'll ultimately ask whether there's a single optimum, so whether all viruses are evolving towards one optimum, or whether there's separate optima, one, say, for the resistant viruses and one for the sensitive viruses. There's a couple of things under the hood that I'm not telling you about, so we have to estimate the ancestral states, and we'll deal with uncertainty for that by running an MCMC chain over those ancestral states and repeatedly fitting these models to a distribution of phylogenies that comes from a beast analysis. And what we find is strong support for a two-state Brownian motion model, where we have separate means for this Brownian motion model for resistant and sensitive, but no restoring force. And what we think is going on is that we're actually probably fitting the wrong model. So what we see is an increase in resistant and sensitive lineages through time. And what I've told you is that we're going to get a steady increase in the binding avidity as the number of previously exposed hosts or humans goes up in the population. So we probably need to include a term in the model where there's some expected increase or some linear trend in the binding avidity that's explaining very likely why we don't have convergence back towards some optimum. And that's a model that we're actively working on right now. The last thing that I want to plug is we're trying to do this for the 2009 post-pandemic flu, but none of these have been typed by the CDC, and we're actually working with the CDC to deploy convolutional learning networks or deep learning to predict the phenotype of these viruses, and so far it seems to be working quite well. We have the Python code to basically allow you to use genomic data in Keras or TensorFlow to build genotype-phenotype maps that we can share soon. And if you're interested in experimental evolution, I am looking for a collaborator that can help me test this in the lab, so in principle we can evolve influenza in different antibody concentrations and ask if the speed of evolution towards resistance actually is faster. And as I mentioned, I am updating my affiliation. I'm moving to Northeastern University in September, and I realize that I'm out of time. Thank you very much. If there are any questions, I'm happy to take them since it's lunchtime, but you should not feel obliged.